BioVivacious. I'm Sebastian. BioVivacious is a YouTube channel dedicated to clear fundamentals of biosciences and make the subject exciting. Today let us focus our attention on glyoxylate cycle. So this is another alternate pathways to carbohydrate metabolism. Now this pathway is looking very similar to the TCA cycle with bypassing a few reactions. We will be looking elaborately at this. Now in plants and in microorganisms, if the fatty acids derived from glycerides, it can be converted to oxaloacetic acid. And you know that this oxaloacetic acid is potentially it is glucogenic. So it can be converted to glucose. So this oxaloacetic acid, it is fed into a cycle known as glyoxylate cycle. So that is how it can result into ultimately carbohydrate metabolism. So therefore let us look at this particular pathway. So remember, it all begins with the similar to TCA cycle. We begin with the oxaloacetic acid and the acetyl CoA, which comes from fatty acid oxidation or carbohydrate oxidation. This will be ultimately converted to citrate. Okay, it is converted to citrate. So the enzyme is citrate synthase. Now, once this is formed, this citrate is converted to isocitrate, similar to TCA cycle. Okay, it is converted to isocitrate. Now, once isocitrate is formed, so the enzyme is aconitase. Okay, we can refer to the TCA cycle. Once isocitrate is formed, now comes the important enzyme, a new enzyme. Okay, remember in TCA cycle, isocitrate, it will undergo two decarboxylation reactions. We are bypassing those decarboxylation reactions. Now we will convert isocitrate, we will cleave by the enzyme isocitrate lyase. Okay, isocitrate lyase will cleave this two. Now the two products that we are going to get, one is succinate. Succinate is, you know that it is part of the TCA cycle. The other product is very important for us. That product is glyoxylate. Okay, see how we are bypassing the TCA cycle. Now this glyoxylate, it is a new molecule. The structure is, okay, this is a structure of glyoxylate. Now to this glyoxylate, see, we want to ultimately make the maximum use of acetyl CoA. Now to this glyoxylate, another acetyl CoA is added. So we are adding another acetyl CoA. So one acetyl CoA is added here, the second acetyl CoA we are adding here. So in a normal TCA cycle, acetyl CoA enters the two carbon atoms and the two carbon atoms would have been removed in the next steps of TCA cycle. Now in this case, we are not removing carbon as CO2. So we have added 4 carbons as acetyl CoA. Now what happens? Now these acetyl CoA, so the CoA will be removed and this will be converted to malate. Malate is also another intermediate in TCA cycle. So this is by another enzyme. That enzyme is malate synthase. So now malate will be converted to oxaloacetic acid, and this is basically the glyoxylate cycle. What is important for us to remember is we are introducing two new enzymes. One is isocitrate lyase, another one is malate synthase. So therefore, by with the help of these two enzymes, acetyl CoA is converted to glyoxylate, okay, instead of converting that as two molecules of carbon dioxide and then combined with another acetyl CoA, and this cycle continues. Now let us see how this contributes towards carbohydrate formation. Let us us summarize what we have seen so far. So therefore, glyoxylate cycle involves two cell organelles. One is the glyoxysome, other one is mitochondria. So let us begin this pathway starting from here, this oxaloacetic acid. So therefore, these are reactions that will occur in mitochondria. So therefore, we will block it like this. So these are reactions occurring in mitochondria. So it begins from here, oxaloacetic acid. Now, oxaloacetic acid, first of all, it will be through with the help of a transaminase, aspartate transaminase enzyme, this will be converted to aspartic acid. Okay, so now the amino group is given by glutamate, it gets deaminated into alpha ketoglutarate. So transaminase is the enzyme. Now what happens is, now it involves the coordination of another cell organelles, that is this. So therefore they are bifurcated. So this is the glyoxysome. Okay, so this is the glyoxysome. Now this aspartate which is formed in the mitochondria, you see it has to be transported into glyoxysome. So once this aspartate is in glyoxysome, exactly the reverse reaction occurs, catalyzed by the same transaminase. So it will convert aspartate into oxaloacetic acid. You see how oxaloacetic acid is made available in glyoxysome. Once oxaloacetic acid is in glyoxysome, the pathway which we have seen in the earlier frame is carried out. So acetyl-CoA is added, citrate synthesis enzyme, citrate is formed. Once citrate is formed, aconitase will act on it, isocitrate is formed, similar to TCA cycle. Now the change occurs. Isocitrate is cleaved by isocitrate lyase. Rather than undergoing decarboxylation reaction, it is converted to succinate and glyoxylate. We have given the structure in the previous slide. So glyoxylate is formed. Then comes another acetyl-CoA is added to glyoxylate, and it, with the help of a new enzyme, that is malate synthase, you will get malate. So with this, in this glyoxysome, two products are formed. One is malate, other one is succinate. These are the two end products formed in glyoxysome. What is the fate of these two products? Succinate transported into mitochondria. You see how the coordination of these two cell organelles are very important for this pathway. So succinate is transported, and succinate will continue with the remaining steps of TCA cycle, converted to fumarate, malate, oxaloacetic acid. Now this cycle continues. What happens to malate? Malate will be transported into the cytoplasm. Malate is now, it can be converted to oxaloacetic acid, very easy, using the enzyme. Okay, here, so you are able to see, using the enzyme malate dehydrogenase. And you know that from our earlier videos, oxaloacetic acid can be a starting material for gluconeogenesis. You see how fatty acid in the form of acetyl-CoA becomes part of how it is contributed towards formation of glucose. So this is what is known as the glyoxylate cycle. Remember, we have bypassed three reactions of TCA cycle. That is from the conversion of isocitrate to alpha ketoglutarate acid, the first step, then to succinyl-CoA, second step, then the third step is succinate. So if the carbon dioxide is not formed, we have bypassed those three reactions, and we have allowed two acetyl-CoA molecules to get inside and ultimately get converted to sugar molecules, to glucose molecules. Let us write a stoichiometry for glyoxylate cycle. So therefore we have allowed 2 acetyl-CoA to enter, 2 NAD plus is added, 1 FAD, okay. So that will result into oxaloacetic acid plus 2 CoA molecules plus 2 NaDH plus 1 FaDH2 plus 2H plus. So this is going to be the overall stoichiometry of glyoxylate cycle. Let us look at the significance of glyoxylate cycle. So there are many points to be added to this. The first and foremost is see the oxidative decarboxylation of pyruvate to acetyl CoA. It is an irreversible process in most organisms. So this has got important consequences. It means that the carbohydrate can readily be converted to fat, but fat cannot be converted to carbohydrate or to protein. So therefore this pathway allows energy to be obtained from fat. So allows energy to be obtained from fat. That is the first significance of this particular pathway. The second significance of this pathway is, you see some photosynthetic anaerobic bacteria, it can directly convert acetyl CoA to pyruvate, okay, with the help of reduced ferredoxin. So, which is a very powerful reducing agent than NADPH. So this pathway allows, especially by providing acetyl CoA. So therefore it can be written, point number 2 can be written. So acetyl CoA is converted to pyruvate, okay, and CoA SH. So what happens is, so therefore ferredoxin, we will write it to ferredoxin like this. So this is the reduced ferredoxin, and ferredoxin, this will be the oxidized ferredoxin. So this is possible because of the glyoxylate cycle. The third significance of glyoxylate cycle is, in plants, fungi, bacteria, they need a large amount of glucose for synthesis of polysaccharides like, cellulose, glucans, chitin. Also for seed germination, extra glucose is needed, and all these is provided by glyoxylate cycle. So especially through the beta oxidant glyoxylate cycle, so additional carbohydrate can be produced. So additional carbohydrate produce. So this is the third significance of glyoxylate cycle. The fourth significance of glyoxylate cycle is very important, that CO2 is not lost. So it is not like the TCA cycle. In this pathway, carbons are not lost as CO2. So this makes a kind of the significance, you understand the significance of this pathway, especially in plants and in microorganisms. Remember, human beings, animal system, we don't have glyoxylate, so therefore this pathway is not operational in man and other animals, because they lack this particular enzyme system. These two enzymes are absent in us, and therefore this pathway is not significant in our system, more significant in the case of plants and in microorganisms.